topic, I thought I'd take some time today and give a little bit of a summary of everything else that's going on in the analysis of PFAS that is not the main focus area, for example, like drinking water. And I will be focusing on market trends in the testing, especially in alternative proteins and phytoremediation. If I can figure out how to advance my slide. Where would I do that with the arrow? I try the arrows. Okay, so in a publication from 2021, the FDA has looked at traditional protein-rich foods. And you can see in one of their results tables that is shown here on the right, the values are not particularly high at the moment of what they found. So like canned tuna, fish sticks are pretty much within the limit, also the limit that we expect to having to hit in Europe. But what was quite interesting, the protein powder concentrate was a bit higher, and that prompted me to look a bit further into alternative proteins. So alternative proteins being proteins, it is important to note that unlike other residues, PFAS are not stored in the fatty tissue. They have quite a high affinity to bind to proteins. And that's why we would expect to find them in quite high amounts in protein-rich foods. They can come in from the production side, of course, but also from food packaging and be enriched in those kind of foods. So food packaging might play a different role in those food matrices in the future. But one point I particularly wanted to highlight is the cell-cultured meat. So when you grow cell-cultured meat, you typically in a setup have meats in a bioreactor, have the tissue cells in a bioreactor, and you change the cell culture media. And one main ingredient in the cell culture media is water. And if you keep changing it and the protein, the muscle cells that you're growing have a high affinity for PFAS, it could act like a solid phase extraction from your cell culture media and actually become enriched in those matrices. So for cell cultured meat producers, water testing will be crucial so they know if they have to do any additional treatment, for example, and maybe water filtration is needed for folks who are producing that kind of foods. SCIEX has done a lot of work in the field of water testing. I just want to highlight a few things. So we have tech notes on PFAS quantitation in seawater, on the SCIEX 7500 system from this year, from Jess, who was just on, by the way. And then there's several drinking water applications on different triple quad systems, which you can see at the bottom of the slide. So for novel foods, at the moment there is missing regulation, but what there is safety data requirements. They differ a little bit between geographies. So I picked an example here. You can see a picture of the PDF that the Singapore Food Agency put out. So these safety data is largely required to bring alternative proteins to the market, or other novel foods for that matter. And you can see that PFAS at the moment are not mentioned in those. So there's a mentioning of mycotoxin and there's a mentioning of pesticides, but we haven't seen anything specifically relating to PFAS. And we would keep pressing the wrong button. We would quite widely expect to see similar regulations for alternative proteins when it comes to food testing, but a few could potentially be different. So the same things, what we would expect is, for example, for both cultured and conventional meat, that people would have to check for vet drugs, either if they're grown in the animal, they might be necessary, or they might be necessary to keep preventing any sort of disease in the bioreactor as well. And other forms of pest management might be necessary there as well. And for plant-based meat, we would expect to typically see the regulations apply to the raw materials. So if you make a bean burger, it would be applied to the raw beans. But you can see that the alternative proteins at the moment is quite a focus of discussion. So in the last meeting of AOSC in August this year in New Orleans, it was put on the agenda on numerous occasions. You can see a little snippet of the agenda on the lower right hand, and the I think the consensus is not quite there of what compounds to test for in these matrices, but the discussion is there. And at the moment, it's at an earlier stage. So researchers are commending the use of established biopharma applications for protein characterization, like post-gestation modification or amino acid sequencing for those. And in the future, we expect to see a bit more detail on the compounds that are being tested for. As alternative proteins or other food, especially the plant-based proteins, are grown in soil, is a logical step to look at how can we make those kinds of food safer. And what people have done actually for a while now, it's not that new. It's just that the publications in research have been ramping up over the last years as PFAS came more into the focus of research. So the problem is when you have agricultural soil contamination, the PFAS end up in food, and they can get into plants in two different ways. So they can either be taken up from the soil over the roots, which is what the remediation efforts would focus on to prevent that, but they can also be deposited from atmospheric particles when they land on the above plant parts of the plants. And then plant-based protein-rich, especially processed foods like bean burgers that I mentioned earlier, have the potential to have quite high amounts of PFAS for that protein binding reason. So we've seen a rise in PFAS testing of soil before and after remediation. Of course, people always want to know how effective was my cleanup, and we've also seen a rise in publication that look into what different plant species that are used for remediation, how well they take up PFAS, how well they can cope with effects of PFAS. And that's used to understand the uptake capabilities of PFAS for specific site cleanups. And when we think about food, or because my job is in food, when I consider PFAS and phytoremediation, I always think about terrestrial plants, so everything that can be used to clean up agricultural soil, but actually the remediation efforts go a lot further. So people also look into the capabilities of how aquatic plants can clean up PFAS from water that is either later used for irrigation or is in the rivers or is in the ocean. And I just picked a few examples to show you how complex phytoremediation is. So here is a publication from a group in Sweden, and they looked at the cleanup of PFAS from a Swedish fire training facility near the airport in Stockholm. I forgot the name. I think it's called Arlanda Airport. And they looked at different species. So they had to look at trees, bushes, and ground cover species. And they found that the maximum concentration is indeed in the foliage, so the leaves and the twigs. And you can see on the right-hand side, it's quite small actually, and then when you present it like this, the different plant species have very different uptake behaviors of different types of PFAS. So you can see one PFAS, the dark green one, which is 6,2-FTSA, has very high uptake in some plants and almost none in the others. Like if you see like the fourth one, for example, but just to show you the it's almost like a relationship between the plant and a specific type of PFAS. Every plant has a specific preferences for a certain type of PFAS, and that makes it very complex. One interesting thing that this publication found, that tree roots, because they have quite a deep penetration, they go much lower than ground cover species, for example, plants that grow in a meadow, they can potentially reach groundwater and not only clean up soil, but also the surrounding water in that area. And there's another example I wanted to share with you, which was recently published. It only came out on the 3rd of November. And that was people have looked at phytoremediation with hemp, which is quite often used for remediation efforts. So they looked at paired hemp and samples and looked at 28 different types of PFAS in the soil and found that the hemp plant can take up 10 of those. The others have not been found. And hemp exhibited a greater bioconcentration for carboxylic acids compared to the sulfonic acids, and was also more preferable for shorter chain compounds compared to the longer chain bonds. Unfortunately, when you look at the percentage of PFAS that have been taken up by the hemp plant is quite low. So 2% was reported in that publication, but the degradation, which the paper used, was hydrothermal liquefaction, and there were some quite good findings, so that hemp resulted in nearly 100% degradation for carboxylic acids and a little bit less good in performance for the sulfonic acids that remained. So the PFAS uptake in plants has been investigated in food plants and in non-food plants. So just a few examples to show you the work that has been done recently in 2023. So this year there was an interesting publication in ferns for site cleanup, but also typical food plants like rice and peas and maize have been investigated, wetland plants and aquatic plants, as mentioned before. And the review I wanted to share with you is shown on the right-hand side. And you can see that the PFAS uptake in plants has quite a big variety. So the plants, this group has looked up the PFAS uptake ranged from 0.2% up to 33% of all PFAS taken up per crop cycle. And they have estimated crop cycles, like how often do you need to let a plant grow to remove PFAS from that site. And it was found that the crop cycles that are potentially needed range between 6 and 233 crop cycles for sunflower, and equally in that range and a little bit higher for hemp. And because this PFAS uptake is so complex, one trend that I've seen recently that I wanted to share is no surprise. AI has been used to model PFAS uptake and different behaviors, and that has been done in an example shown here on the left-hand side for animals, because it's important to understand the toxicokinetics, as they are currently not known for all PFAS because there are so many. And one of the problems is that there's very long half-lives that have been observed in some cases. And it's very crucial to know the chemical-specific half-life, because that's necessary to make a reconstruction of the exposure and also to extrapolate from toxicological studies. So people look at how PFAS behaves in different types of animals, including humans, but also how PFAS behaves in the environment. And the review article I wanted to share also from this year looked at the prediction of PFAS uptake and the various factors that can affect it. And I only picked a few, but there were many more mentioned. So it can be affected by the organic carbon contents of the soil, the pH, of course, the PFAS concentration in the soil, but also root protein contents. Makes sense. If you have more protein you can bind more. And what I found quite interesting is the soil temperature, as it's directly correlated. That might become interesting in the future years with regards to climate change. So some of the challenges of phytoremediation are that PFAS uptake in plants typically is quite slow. It depends a bit on the growth rate, and it often PFAS, they're unhealthy for humans. It's no surprise they also have phytotoxic effects, which means they are having negative effects on plants. So that's why it's important to identify PFAS hyperaccumulators that have quicker growth rates and potentially also, ideally, an enhanced resistance to PFAS toxicity, so they can accumulate more and it doesn't matter so much for them. And it's also important to find or to work on genetically engineered crop plants that have lower PFAS uptake that might become more important for food production in the future. So there's a lot more research needed, especially when it comes to relationships of like plant-associated microbial communities or other molecular processes that have to do with mainly with transport proteins in the routes for PFAS uptake. As we talk about alternative proteins, I wanted to highlight one thing that I found quite interesting. When we eat more, for example, plant-based proteins in our diet, it could have exposure. It will change the exposure to certain chemicals. That always happens when you change your diet. And the Trilogy Lab in the US has found something very interesting. They detected quite high levels of ochratoxin A, a mycotoxin, in soy-based baby food. In some cases, that was extremely high. But they also mentioned that in a study from Germany, they found that vegans had strikingly higher ochratoxin A levels in their blood than the comparison group. So I put a picture of a soy latte here, which is something maybe a few people can relate to. And the diet of the group of the vegans differed primarily in the significant higher consumption of legumes. So as legumes are also typically the main ingredients for plant-based, high-protein alternative products, just as burgers or protein shakes or bars, they therefore also pose a higher risk for PFAS contamination. And I'm off to my last slide. So when we talked about nutritional changes and PFAS being found in most of our food products these days, I wanted to point out that it can not only end up in us, but it could also end up in other people via blood donations, for example. So a study from Australia looked at PFAS levels in firefighters, which are naturally have a high exposure towards PFAS from the firefighting foams. And they found that after a single blood donation, the mean serum level of perfluorooctane sulfonate, which is PFOS, were reduced from 2.9 nanogram per mil to 1.1. So that is a significant difference here, and similar changes were seen with other PFAS. So you can, after you've eaten it, you can reduce your blood PFAS load with blood donations. It's just there's a risk for the recipient. It has to do with weighing up the risk. If you need a blood transfusion, you probably don't need to worry about PFAS exposure in that moment. But it's an interesting application, I find, as PFAS bind, especially to albumin across species, but very important for humans here. They could accumulate and it could be transferred to patients. And it could also happen that they either leach from the blood bag material into the product which people receive, or it could be that they could be absorbed onto the container. So if anybody does any research in that area, I just wanted to say I would love to hear about that. I haven't found much, but I wanted to include that in my talk as well. So we're coming to an end. In summary, the monitoring of PFAS in environmental matrices is highly interconnected. We have new questions arising the whole time. Technology gets used in new ways. SCIEX is watching these trends very closely, and we will try to keep you updated. And the easiest way to get updates from us is if you follow us on LinkedIn. So if you like, you can find the SCIEX Food Environmental Testing Solutions. And that is all from me.